Hi guys, today we will be looking at the control of blood water potential. Mainly we will focus on the structure and function of the nephron, so the working unit in the kidney. We'll start with the structure and naming the processes that are taking place in different parts of the nephrons. So start with drawing the first part of the nephron, which is called a renal capsule. So we've got the renal capsule here. And the process that takes place in the renal capsule is the formation of the glomerulus filtrate by ultrafiltration. Okay. Following the renal capsule, we can then go further to the next part of the nephron, which is called the proximal convoluted tubule. Within this section, this part of the nephron, the second process is taking place, which is called the reabsorption of glucose. Here we will also be looking at the reabsorption of sodium and some reabsorption of the water in a second. Following then the proximal convoluted tube, we've got the next part of the nephron, which is called the loop of Henle. Okay, so that's the third section here, loop of Henle. In the loop of Henle, what we need to pay attention to, as we can see, we've got two limbs. One of the limbs is going down, as we can see it over there, and that loop we call descending. The other loop on the other side going up towards the distal convoluted tube, we call that ascending. Okay. We will be looking into details what happens in those loops. So there will be adaptations of both descending and ascending limbs, and we will look into that in a few seconds. Most important thing that happens here in the loop of Henle is reabsorption of water. And of course the maintenance of the concentration of sodium ions. And this probably will be the most difficult process to kind of understand and memorize for the function of the nephron. Finally, going up through the descending limb, we are arriving at the fourth section of the nephron, which is called distal convoluted tube. Which then further takes us to the final part of the nephron called collecting duct. Both of those sections, fifth and fourth, on the diagram, are involved in the reabsorption of water. Okay, so to summarize the main structure of the nephron and the main processes that are taking place at them, we've started with the renal capsule. Renal capsule is the first part of the nephron where the ultrafiltration, so the initial formation of the glomerular filtrate, is taking place. After the renal capsule, we can see the proximal convoluted tube, which is responsible for the reabsorption of glucose. We will also see some reabsorption of sodium ions and water. After the proximal convoluted tube, we've got the loop of Henle, which is made of two limbs, descending, going down, and ascending, going up. This part of the nephron is responsible to maintain a gradient of sodium ions, and also here we will see some absorptions of water. Going up, we are reaching the distal convoluted tube, which leads us to the collecting duct, and both of those parts of the nephron are responsible for the further reabsorption of water. In terms of the location of the nephron in the kidney, we can find the renal capsule, proximal convoluted tube, and distal convoluted tube in the cortex, whether the loop of Henle and the parts of the collecting duct are located in the medulla. Okay, in the next parts of this quick revision session, we will be looking at the processes that are taking place in each of the parts of the nephron. So we will move on to the renal capsule. As we've said, in the renal capsule, the process of ultrafiltration is taking place. The renal capsule, the first part of the nephron, is accepting a lot of things from the glomerulus. So from the many blood capillaries that are delivering towards the nephron, towards the kidney. The formation of the ultrafiltrates is quite similar to the formation of the tissue fluid. So if you want to quickly recap on the formation of the tissue fluid, that would be great, because then you will understand the ultrafiltration much better. Obviously, the blood vessels that are carrying important molecules towards the renal capsule are coming away from the heart, and if doing so, they will meet the nephron, the renal capsule, with the higher hydrostatic pressure. If you remember properly, coming out then, following the processes that will take place, we will have lower hydrostatic pressure. The most important thing that we need to pay attention to is now how the difference in the hydrostatic pressure allows for the filtrate to be formed. Obviously, within our glomerulus, in the blood capillaries, we've got many important things that we wish to get back to our body. So it could be amino acids, could be glucose, could be water, could be urea, any small molecules. So any of those small molecules will be allowed to come out of the glomerulus and squeeze through the cells that are making the basement of the renal capsule. Those cells are called podocytes. The cells are actually forming here a great deal, giving the renal capsule a great deal, because as you can see, there are gaps in between those cells. The gaps allowed for those small molecules, amino acids, glucose, water, urea, and so many more, to pass through the podocytes to form the initial filtrate, which we called the glomerular filtrate. Okay, if you remember properly from year one, within the blood vessels, We will also have the larger molecules, for instance, proteins. Looking at the size of this protein, hopefully you can gather that proteins are too large to become the part of the glomerular filtrate. They will not leave the glomerulus, they will remain there. So, if proteins are staying within the glomerulus, obviously the proteins are causing the difference now in the water potential. So, due to proteins, we will now experience lower water potential in the glomerulus, hence some water might obviously remain there as well to cope with that difference in the water potential. So as the product here of the ultrafiltration, we have received a glomerular filtrate. How was that done? One more time. The blood vessels arriving away from the heart towards the nephron are causing the difference in the hydrostatic pressure. Coming towards the nephron, we've got higher hydrostatic pressure. Going away from it, the hydrostatic pressure now it's lower. This is due to the process called ultrafiltration. In the formation of the glomerular filtrate, the small molecules such as amino acids, glucose, water, urea will be squeezed out of the glomerulus to form the filtrate, and they will pass through the cells, making the basement of the renal capsule called podocytes. Squeezing through them, they are forming the initial glomerular filtrate. Within the glomerulus, of course, we're leaving behind the large molecules such as proteins because they are too big, too large to pass through the blood vessels. Hence, they will lower the water potential. And of course, if you remember from year one, the lower the water potential, okay, more osmosis towards will happen, which is the movement of water from higher water potential to lower water potential. So that's the ultrafiltration in the renal capsule. So this glomerular filtrate, of course, has here the glucose. Glucose is important to us because it's a substrate for the aerobic respiration, and our body will want to recover glucose. The recovery of the glucose, the reabsorption of the glucose, happens in the proximal convoluted tube. So, of the renal capsule that we were looking at a second ago, we've got the proximal convoluted tube. Imagine this is the lumen of the proximal convoluted tube. Here we'll have the epithelial cell within the proximal convoluted tube. So epithelial cell of the proximal convoluted tube. And of course, as we've said, that important molecule glucose, we want to reabsorb back. We don't want to lose it with the urine. We want to get it back to the to our system. We want to get it back to the blood capillaries. How is it done? Again, really, it's a really synoptic concepts here, exactly the same as you would want to describe the co-transport, which you've learned about in year one. So within the co-transport, obviously, we want glucose, and there would be movement of sodium ions as well, to get into the blood capillaries. You want to reabsorb it back. So how this is done? Within the epithelial cell of the proximal convoluted tube, we've got sodium ions. Sodium ions are actively transported from the epithelial cell into the blood capillary against the concentration gradient. Transporting sodium ions away from the epithelial cell of the proximal convoluted tube lowers the concentration of sodium ions here, meaning we have now maintained diffusion gradient for the sodium, because in the lumen we've got higher concentration of sodium ions than in the epithelial cell, because of course active transport of those took place. So what will happen now? We will see co-transport, which is movement of two different molecules from the lumen of the epithelial cell into the epithelial cell. So we've got glucose and sodium ions. We'll use different colors for those now. Okay, and they are now coming through the co-transport proteins, through the carrier proteins towards the epithelial cell. By doing so, of course, we're now increasing the concentration of the glucose in the epithelial cell. Having higher concentration of the glucose in the epithelial cell allows it for the facilitated diffusion from the epithelial cell into the blood. Okay? To finish off this diagram, we obviously need to now come back with the ideas from year 1. So we did say that the first process that takes place is the active transport of the sodium ions from the epithelial cell into the blood. This transport, of course, requires carrier protein, and it also requires ATP. This transport, active transport of the sodium ions from the epithelial cell into the blood, allows to maintain the concentration gradient of the sodium ions now in the epithelial cell, because comparing to the lumen, now we've got lower concentration of sodium ions than in the lumen. Hence, co-transport of glucose and sodium ions is allowed. Again, to be for the effective transport, we require here carrier protein. Remember, the carrier proteins that will be involved in this process will be different, will have different 3D tertiary structure, because we are transporting through the facilitated diffusion glucose and sodium ions, which of course are different. Hence, following the process, now we are achieving higher concentration of the glucose in the epithelial cell, which we are one step away from recovering the glucose. Having higher concentration of the glucose in the epithelial cell allows it to now be transported by facilitated diffusion using carrier protein into the blood down the concentration gradient. And that's the whole point here, of course, of the proximal convoluted tube. It's nothing else than the co-transport, which explains how the glucose is reabsorbed back from the nephron, from the proximal convoluted tube into the blood. However, what we need to pay attention to here is the fact that, of course, having importing the sodium ions and the glucose into the blood capillary, we now, by doing so, lowered water potential in the blood capillary. Having lower water potential in the blood capillary, obviously, can be sorted by further osmosis of water. Hence, osmosis will move from the higher water potential in the epithelial cell in the proximal convoluted tube into the blood vessel, into the blood capillary by osmosis, so movement of water. Hence, when we started looking at the structure and function of the nephron, we did say that the main job, of course, of the proximal convoluted tube will be to reabsorb glucose. However, as we were doing so, glucose and sodium ions lowered the water potential in the blood capillary. Hence, we needed to reabsorb water as well to, of course, overcome that difference in the water potential. So now, if we're looking at our glomerular filtrate, it's missing some glucose and sodium and water because they has been removed from the initial glomerular filtrate. Following the proximal convoluted tube and the process of reabsorption, we are aiming now to arrive at the loop of Henle. Remember, loop of Henle is the part of the nephron that is located in the medulla. So far we were working in the cortex, now we are in the medulla. So this is loop of Henle. Okay, the third process that happens in the loop of Henle is, of course, the maintenance of the sodium ion concentration and some absorption of water as well. We did say that the loop of Henle, it's formed of two limbs. One limb here, it's called descending. And the other limb called ascending. Those limbs got different jobs, hence they will have different characteristics. Descending limb, we need to remember, it's permeable to water. And what many of the students don't understand what permeable to water means, or what the fact of being permeable means, it's actually allowing the water to come out. Being permeable to water, it's one characteristic of descending limb. The other characteristic of the descending limb, it's the fact that it's impermeable to sodium ions. So you can guess quite easily here, if permeable means it's allowing things to come out, impermeable means that the things can come in. Looking at the ascending limb, because obviously biology it's not that straightforward, just to confuse you. In the ascending limb, we've got different scenario. The ascending limb is impermeable to water. What does that mean? Now you should be confident with that. It means the water can come in. However, it's permeable to sodium ions, which then makes them being able to come out. Okay, and this is how we're going to look at the maintenance of the sodium ions and some movements of water in the loop of Henle. So I'm going to move on to the clean slide because there's a lot of information happening here, and draw our loop of Henle one more time without the labels, so I've got the space to explain what happens in each of those limbs. Okay, and remember, we are now in the medulla. Okay, this part of the nephron here is in the medulla. We were in the cortex before. So just to remind you here, we had two limbs descending, going down, permeable to water, impermeable to sodium ions, and ascending limb going up, which is impermeable to water but permeable to sodium ions. So we are going to look now at the processes that are taking place in the loop of Henle that allows the maintenance of the sodium ion concentration. So I'm going to start the story of the maintenance of the sodium ions concentration with the ascending limb. So one more time, with the ascending limb, we say it's impermeable to water, means that the water can only come in, And also it's permeable to sodium ions, which allows them to come out. And that's what we will start looking at from. We will look at the movement of sodium ions. Sodium ions are actively transported out. Sodium ions are actively transported out from the ascending limb. Because, as we've said, ascending limb, it's permeable to sodium ions, hence they can come out. Quite similarly to what we observed in the proximal convoluted tube, the movement, of course, of the sodium ions out, has an effect. And that effect will have on that space between the limbs and the space between the loop of Henle and the still convoluted tube. So, influx of those sodium ions into those spaces now lowers water potential. Okay, so you got lower water potential in those spaces here. So now we want to, so that's the second point on our maintenance of the sodium ions in the loop of Henle. So we actively transported sodium ions out of the ascending limb. Just one more time, remember, ascending limb was impermeable to water, permeable to sodium ions. Having actively transporting them out, we've lowered the water potential in the spaces in between. So, logically thinking, we now want to deal with this water potential. And we've got two options here. Option number 1 is to get some help through the osmosis, or option number 2, which will be more logical as well, is to try and get rid of those sodium ions. And to remind you, we can go for those two options. Why we could do so? Because descending limb, it's permeable to water, going to allow water to come out. Also, it's impermeable to sodium ions, which is allow them to come in. And that's what we can do. So we will start with the easy process, logical process, to get rid of those sodium ions that lower the water potential. We can just transport them now to the descending limb, because as we've said, descending limb is impermeable to sodium but permeable to water. Having been able to do so, we can now move the water out. So the second option that helps us out with the maintenance of that water potential is the fact that the water can come out of the descending limb by osmosis. Why is it possible? Because we had higher water potential in the descending limb, and remember, lower water potential in the space between them. So we've sorted out the left-hand side of our scenario. However, remember, the lower water potential was also created in the space between the ascending limb, distal convoluted tube, and collecting duct. Those two parts of the nephron will further help out with the maintenance of the water, and it will allow the reabsorption of the water, so the water will come out again from higher water potential to lower water potential by osmosis. Okay, so our filtrate, glomerular filtrate, that has been, if we remember, initially formed in the renal capsule, went through all those stages, the proximal convoluted tube, through the loop of Henle, distal convoluted tube, finally collecting duct to produce our urine. Also, what happens in the distal convoluted tube if we are dehydrated, same happens in the collecting duct. It's of course the help from the from the hormones. ADH hormone, antidiuretic hormone, could be released. The osmoreceptors in the brain realize that there is not enough water, now the body is dehydrated. ADH hormone will be released. What the ADH will do, it will make the walls of the collecting duct more permeable to water. So doing so by making kind of holes in the collecting duct, which we call aquaporins. So the water can be reabsorbed even more. Okay, what we need to remember here about the job of ADH is to further, of course, help out with the reabsorption of water in the situations when we are dehydrated. Most of the questions that you will see on ADH, it's made on the it's asking you about the volume and the concentration of the urine. So of course, if you are dehydrated, the concentration of the urine will increase. However, the volume will decrease, and this is because more water has been reabsorbed, decreasing the volume of the urine. There are many more past paper questions that you need to attempt now to become confident with the whole process of the formation of the urine, of the control of the water potential. That's everything. Make sure you will make your notes, have your poster, use the model answers, 5 markers, and attempt past paper questions to look at the patterns and be confident when it comes your turn on the exam.